So the next pathway we'll talk about then is the Krebs cycle. We went through the glycolytic pathway and we talked about the transition step. So the Krebs cycle, also known as tricarboxylic acid cycle and citric acid cycle, is going to take in that acetyl coenzyme A molecule right here. The coenzyme A acts as a carrier, and it just basically, it taxis the acetyl group. The acetyl group is dropped off, and it binds to a 4-carbon molecule called oxaloacetate. An oxaloacetate, or OAA, which is oftentimes how it's written, bound to that acetyl group produces citric acid. So now we have the citric acid molecule that is going to be oxidized back to the oxaloacetate. So we're going to basically oxidize the entire acetyl group. So there's going to be nothing left of that original glucose molecule. So each acetyl group has 2 carbons. Each acetyl group has 4 hydrogens. And so we have to have oxygen available to bind to the carbons to produce, for each acetyl group, 2 carbon dioxides. And we also have to have the different coenzyme carriers that are going to get reduced when we oxidize that acetyl group. And so we have, for each acetyl group, we have 3 NAD that gain hydrogens. Sorry, that's an H. And we have 1 FAD that gains a hydrogen. And since we have a total of 2 acetyl groups, these numbers double, which is where we get, okay, I don't know why this is weird, which is where we get, this should say 4CO2. Oh, sorry, oops. That's erased. And then we have 2 ATP, and I haven't talked about that. I will in a second. 6 NADH and 2 FADH. In this process, a small amount of ATP is produced via substrate level phosphorylation. Okay. And so for each turn, we produce 1 ATP, so a total of 2 ATP is produced. And at the end, we end up with this same oxaloacetate compound that we had in the beginning. So we always start and end in this cycle with oxaloacetate. Then we have the pentose phosphate pathway, which is that 3rd pathway. This pathway also breaks down glucose, but in this pathway, we're going to break glucose down so that we can produce different materials for biosynthetic pathways. So we're going to use NADP as our reducing power. We're going to oxidize glucose and produce NADPH from the oxidation of the glucose. NADPH then can move into different cycles and produce larger organic molecules. We also are going to utilize ribulose 5-phosphate, and we can produce ribulose ribose 5-phosphate, erythrose 4-phosphate, which are both utilized in different biosynthetic pathways. We talk about this one in photosynthesis. In our last step of aerobic respiration, we look at the electron transport chain. So the ETC, or electron transport chain, uses the reducing power of NADH and FADH to produce ATP. Now, we produced NADH and FADH in glycolysis, transition, and the Krebs cycle. So the electron transport chain then produces a proton motive force that drives the synthesis of ATP. So think about if you have a membrane and you have a bunch of protons on one side and very few on this side, they're going to want to move from where they are to where they are until we're at equilibrium. They move through a protein pump called ATP synthase. And as they flow through, they catalyze the reaction of ADP, to ATP. Okay, so this was proposed in 1961 by Peter Mitchell, and initially nobody even gave it any thought. He did years of research. He funded his own research, and in 1978 he received a Nobel Prize. It is now called chemiosmotic theory. The electron transport chain then is found within the cytoplasmic membrane for prokaryotic organisms, and it's found within the inner mitochondrial membrane of eukaryotic organisms. So this occurs in the mitochondria. It releases energy that drives the production of ATP through an enzyme known as ATP synthase. And so this is the electron transport chain of the mitochondria. You have complex 1, in which NADH drops off its hydrogens. And then you have complex 3, where FADH drops off its hydrogens. And you have complex 2, cytochrome C, complex 4, where the electrons are shuffled. So hydrogens are pumped from one side of the membrane to the other, and the electrons are shuffled from one carrier to another carrier, okay? In prokaryotes, there's some differences within that mitochondrial, or not mitochondrial, within that electron transport chain. Well, first, it's found within the plasma membrane of the prokaryotic organisms, and then we have some different enzymes that are utilized, and these different enzymes are going to shuttle the electrons around, just like the electron transport chain in the mitochondria, but there's different versions, and this allows for higher or lower oxygen levels, okay? Because think of E. coli, they are constantly moving between aerobic and anaerobic and everywhere in between conditions, because they can live in your gut, which is an anaerobic environment, but they can also come out and live in an aerobic environment. So anaerobic respiration in E. coli harvests less energy, but it still produces more energy than fermentation, which we haven't really talked about. In this case, in anaerobic respiration, they're going to use a different type of final electron acceptor. So in this case, they are going to take nitrate and convert it to nitrite. Sulfur reducers might take sulfate and convert it into hydrogen sulfide. So here is the prokaryotic electron transport chain. So here again, we have where NADH is going to move and FADH. But instead of complex 2, we have succinate dehydrogenase, we have ubiquinol oxidase, And we have different mechanisms of moving those hydrogens. So we have ATP synthase in the aerobic mechanism, but we have an active transport mechanism, a pump that pumps materials across as well. So when we look at aerobic respiration and we look at the theoretical maximum yields, the glycolytic pathway produces 2 NADH, and each NADH is going to give us in aerobic respiration 3 ATP. So for each NADH produced, we gain 3 ATP. Because glycolysis gave us 2 NADH, we get a total of 6 ATP. Transition also gives us 2 NADH, so we're going to get another total of 6 ATP. The Krebs cycle, this is tricarboxylic acid cycle. I'm going to put Krebs over here. They give us a total of 6 NADH, which gives us 6 times 3 is 18 ATP. For each FADH, we gain 2 ATP. So because we produce a total of 2 FADH, 2 times 2 is 4, so we get 4 ATP. So if we look and add these up, we get 6 plus 6 is 12, plus 18 is 30, plus 4 is 34 ATP. This is via oxidative phosphorylation or via the electron transport chain. So ETC produces 34 ATP as their theoretical maximum, okay? But the entire process gives you approximately 38 ATP because you gained 2 ATP net from glycolysis and you gained 2 ATP net from the Krebs cycle. So you get 4 ATP there plus the 34 that you had. So that's 38 ATP total as a theoretical maximum. And that's where this number comes from. Now, in eukaryotic organisms, the theoretical maximum is 36 ATP. It takes energy to move materials from the cytoplasm into the mitochondria. So you're utilizing more energy during the process, which means you're not going to have as much of a gain at the end. And so this is that theoretical maximum of ATP production, okay? And so this actually is a video that goes into the different steps. It's a good video you can watch. You can click on it and just, watch the video, okay? Sorry, that's what I want to do. All right. Oh, that was a long video. That was 13 minutes. I'm going to stop here and we'll get into fermentation in our next video, okay? Bye.